In this lecture, we will discuss about how a protein folds from the sequence to 3D structures and how long it will take to go from the sequence to the structure, that is called the protein folding rate. In the last class, we discussed about protein stability, specifically on stability upon mutation, right. What did we discuss in the last class? Right, we discussed about a database, which database deals with the thermodynamic stability? Protheum, right. So, using the Protheum database, right, we try to analyze the important factors which influence the stability of the mutants at different locations, right. We related the physical chemical properties on one side and the experimental stability, right, on the other side, and we try to relate using correlation coefficient. So, we identified that some properties, right, which are important for stability. For example, in the buried mutation, which property is highly correlating? Right, the hydrophobicity, right, hydrophobicity has high positive correlation with stability. But if the mutation is at the surface, right, then what did we observe? Inverse relationship, right, that is called the inverse hydrophobic effect. That means the change in increase in hydrophobicity will decrease the stability. Then we try to predict the stability upon mutation, right. So, we discussed about various methods. So, the one is the average assignment method, right. How the average assignment method works? Then we will get the values for all the mutants. How many combinations, how many mutations? 380 mutations, right. Then for new mutation, you assign the value from the table, that is a kind of lookup tables, right. Then we can evaluate the sensitivity, specificity, accuracy, right, and correlation and so on. So, then we discussed about the inclusion of the sequence information or the structure information, right, with the different neighboring residues or the surrounding residues, right. And then we discussed about the potentials. There are two types of potentials, right. One is the distance potential and the torsion potentials, and we combine these two potentials to predict the stability change. These are from the structures. If you predict with the sequence, right, we derive several rules, right, from the known information regarding the mutant residue, mutated residue, right, as well as the residues which are neighboring the sequence. We derived set of rules. Then we use the rules for predicting the stability, whether this is stabilizing or is destabilizing. So, we discussed about the structure prediction, right, from the unfolded state to the folded state, right, and then we discussed about the stability. Today we will deal with the folding rate. So, what is the folding rate of a protein means, right? It is the measure of slow or fast folding of a protein from its unfolded state, right, the random coil conformation, to its the native 3 dimensional structure, right. How long a protein will take to fold into a native 3D structures. Why it is important? Because this folding rate helps to understand the kinetics, which is related with several pathologies like the prion diseases, Alzheimer's disease and so on. So, it is very important to understand the folding rate of any particular mutants as well as for the mutants, right. You can see for proteins as well as what will happen if you mutate a specific residue. And the predicting the folding rate, right, is also a challenging task similar to protein folding problem. Right. What is protein folding problem?
Final protein folding.
Right, you can disappear in the native conformation of protein from its sequence, like still it is a challenging. So, next folding rate is also important to see how a protein can fold from the unfolded state to the folded state regarding the time. So, here show an example, okay, here this is amino acid sequence, right, it takes time to fold a specific 3D structures. So, these folding rate vary over 8 orders of magnitude, right. You can see these folds from microseconds to an hour. So, you get two examples, one is a streptococcal protein, here the rate is 2 into 10 power 5 per second, this is very fast folding, and if you see the tryptophans in this, right, it is very slow folding, it takes 1 into 10 power minus 3 per second. So, now the question is how fast a protein can fold. So, I will discuss with the 3 different types of examples. For example, if you take the biomolecular interactions, right, for 2 different proteins or the protein and DNA, so whatever, if they want to make a complex, you can see the upper limit that is determined by the rate at which the reactants, what are the molecules which are involved in the reaction, they come close together by diffusion. So, how much the rate they take. Then if you talk about the folding, here you can see the rate of folding is the rate the polypeptide collapse, right, to form the structure. So, from this you can see. If you take one example, say cytochrome C, right, here the minimum time for folding is about 1 microsecond. This is comparable to the hairpin loop or the helix formation. So, it is about 1 microsecond. In this case you can see the rate, right, for the folding is about 1 per microsecond. Now, I will show a few examples like arc repressor is 10 power 4 per second and lambda repressor it is 5 into 10 power 3 per second and so on. These are for the proteins. Likewise, you can also accelerate or decelerate the protein or increase or decrease the folding rate, right, by amino acid substitutions, like what we discussed about the stability, right. By introducing a mutation we can enhance the stability of proteins, right. So, we identify the mutants which can enhance the stability and we can design proteins. Likewise, in the folding rate we can identify some mutations which can accelerate the folding process. In this case they will go to the proper folding, because if it takes more time then this may end up with this misfolding, right, due to aggregation, right, and some other aspects. So, you can get some diseases, right. This is the reason why we need to see the particular protein which folds into a particular time, right, to stable 3D structures. So, how to measure this folding rate? So, there are various measures to get this folding rate, mainly the staff flow CD and the florimetry. This will give you the rate, right, at different concentration. For example, here the x axis we give the different concentration of the guanidine hydrogen chloride, and the y axis we show the observed rate, right. So, you can see the concentration 1 to the different concentrations up to 10 molar, and if you see this is the folding region and here this is the unfolding region, and we extrapolate to 0 to get the folding rate or unfolding rate at 0 concentration, right, because to get the uniform data for different proteins and mutants, right. We extrapolate here in the folding region, it goes to here. So, here now this is the folding rate in water. Likewise, because here we extrapolate this one, right. Likewise, extrapolate the unfolding region here. So, finally, we get here this is the unfolding rate, right, in water environment, right. This can be analyzed using this chevron plot, right. Now, once we have the data, right, for doing the bioinformatics analysis, right, we require a sufficient number of data and reliable data, right. So, it is very important to construct databases, right. On these aspects, right, first they started to develop few databases. So, one is called the protein folding database. So, another one is after few years they developed kinetic DB, but unfortunately both the databases are not available at the moment. The folding database they try to establish using a collaboration among different experiment lists. They assign the different proteins to a different experiment list to get the data at same concentrations, right, and then they compile all the data and then develop this database. Now, how can we get the data? There are various aspects to get the data. Either we can get from the literature or you can see other databases. For example, the folding race, this will deal with the mutants. To get the folding rate upon mutation, so you can use this folding race, right, this is server cum database, right. So, that is available in our website, right, to get the folding rate upon mutations, right. You can get download the data and you can use it for the analysis. So, for the data for the proteins, right, I listed some of the papers which give the data, actual data we can get from this literature. So, in 2005, right, the Maxwell they formed a consortium and they tried to see the folding rate at same concentrations, right, or same experimental conditions. Earlier days we get the data for the different experimental conditions, because different groups they used the proteins at different conditions and they measured the folding rates and they published literature. So, same groups they assigned this temperature, pH and conditions, they asked them to get the folding rate. In this case you get the data for different proteins at same concentration. This is how they published this paper in 2005 with the same experimental conditions. Then currently there are several other literatures and reviews, right, they discussed about the folding rates of proteins, and currently we get more than 100 proteins, we know the folding rates of the proteins. So, we have this data, then we can analyze, right, what factors or what influence the proteins to fold fast or to fold slow, right. Earlier we discussed about the different types of interactions, also they are influenced with different structure classes. What are different structure classes we discussed earlier? All alpha proteins, all beta proteins, alpha plus beta proteins, as well as alpha beta proteins, right. So, what is the difference between all alpha and all beta? Predominantly alpha and predominantly beta, which interactions are dominant in all alpha and all beta? Medium and short range interactions, right, they influence the all alpha proteins, and the long range interactions are dominant in the all beta proteins. So, if we look into this, which type of proteins fold fast, which type of proteins takes time to fold?
All alpha
All alpha proteins
will fold fast.
fold fast, right, because it is mainly medium range interactions, short range interactions, they fold fast. Okay, now I give you the one examples. So, these are the example for the all alpha proteins, right, you can see here, and these for the all beta proteins. All these proteins are two state proteins and small proteins. What is the meaning of two state proteins?
Either have unstable major conformation.
Right, we have two states, one state the native structure and the other state the unfolded state or denatured states, right. So, you can have the unfolded state to the folded state, there is no intermediate. So, several proteins they fold perfect two states, and also that small proteins we know the experimental data for the folding rates. So, they can also have fast folders and slow folders even if all the proteins are two state proteins and these proteins are small, about 100 residues. So, now I show the values for the all alpha proteins, all beta proteins, and if you look in the all alpha proteins, this is the logarithmic of this folding rate ln kf, right. So, this is very high. So, you can this the rate is 9.6 right per second logarithmic of this. We take the all beta, so we can the value is 1, 2 as well as the -1, right, because that is it takes time. So, if you look into these numbers, generally you can say that all alpha proteins fold faster than all beta proteins, but that is not very strict, because sometimes even all beta proteins can also fold fast, because the one CSP the cold shock protein it folds at 6.98, right, similar to this one. But generally you can say that all alpha proteins fold faster than all beta proteins. So, we discussed earlier all alpha proteins are influenced with the short and medium range interactions, right, and the all beta proteins with the long range interactions. Which residues influence long range contacts and which residues influence medium range contacts, right, I will tell you. So, if you see the fast folding proteins one group and the slow folding groups another group, and see the amino acid compositions or the preference or the contacting pattern, then we could see some patterns for the slow and fast folding proteins. For example, if you see the polar residues, so asparagine, glutamine, lysine, serine, these residues, right, are predominantly present in the fast folding proteins. If you see the difference between slow and fast fold, right, this is 5.2 and fast folding it is 9.5, right, it is about 2 times. Likewise, these residues they are occurring highly in fast fold proteins, right, compared with the slow fold proteins. Then also if you see these medium range contacts, right, you can see the contact between the polar residues are dominant in the fast folding proteins, right. This earlier we discussed about this interactions, contacts as well as the composition. So, that agrees with the experimental data for the fast folding proteins. Likewise, if you see the slow folding proteins, what do you expect? Which residues should be dominant? Hydrophobic residues, right. Then about the contacts, long range contacts made by these residues, like this is what we did, right, with the. So, higher occurrence of alanine, cysteine, glycine and leucine. So, these residues are predominantly occurring in the slow folding proteins than fast folding proteins. For example, cysteine there is 3 times, right, here also you can see the difference, right, in the case of slow folding proteins. Then the formation of this hydrophobic core involves long range interactions, right, this is how it slows down the folding process. Then we see the contacts in the slow folding proteins and fast folding proteins, you can see the long range contacts and slow folding proteins, right, they are mainly influenced with the hydrophobic pairs. See alanine alanine, tryptophan leucine, alanine glycine, right, cysteine tyrosine, right, you can see the contacts between the hydrophobic residues are more in the case of the slow folding proteins compared with fast folding proteins. So, we see the slow folding and fast folding proteins, generally all alpha proteins fold faster than all beta proteins, and if you classify into 2 groups, right, which one is fast folding, which one is slow folding, right, and see the preference of residues, right, mainly the polar residues in the fast folding and the non-polar residues in the slow folding proteins. Likewise, the interactions or the contacts, right, mainly the slow folding proteins are dominated with the long range contacts using these hydrophobic residues. Okay, now the question is whether we are able to predict or relate these folding rates with any of the parameters, right. We discussed about various parameters obtained from sequence or structure. What are the various structure based parameters we discussed earlier? structure based parameters, accessibility, contact order, long range order, hydrophobicity, right. So, various parameters, right. So, also if you see the slow and fast folding proteins, the contacts are very important. So, we convert the contacts, right, the 3D structures we convert into contacts using in the form of contact maps, right. You get the contact maps, and from these contacts you deduce various parameters like contact order, long range order, and so on. So, now we will see how these contacts, right, can be used for predicting or understanding protein folding rates, whether there is a relationship between the contacts and the folding rates, right. Basically yes, right, because if you see the more number of long range contacts, that will slow down the process, right. Let us see. So, what is the contact order we discussed earlier? How to define the contact order? This is given as delta sigma Sij by L and N. What is delta Sij? Sequence separation between the contacting residues. So, we construct a sphere of radius 6 angstrom and look at the contacts, right. Then check the distance separation, and that will give delta S. What is L? A total number of residues, that is the length of this protein, and N is total number of contacts, right. So, we do it for each residue to get the delta Sij, add up together and then normalize with the length and the number of contacts, right. So, here I show the data. For example, if this is the sphere 1 and 7, delta S is 6, likewise for all the residues you compute, right, get for the T1, then you repeat it for all the residues in the protein, right, then we will get the value. So, then we relate that value with the folding rate. If there are more long range interactions, what will happen to the contact order? So, more long range interactions means what will happen in this equation? This will be high, because delta S will be high. If this is high, then what will happen in the contact order? That is high, because directly proportional, right. So, in this case you get the contact order is high. If the contact order is high, what do you expect the folding? Folding is slow, because more number long range contacts, then there is folding is slow. For example, there are more short range interactions or more medium range interactions, then compared with the long range contacts. So, you can see contact order is less, because delta Sij is less, right. Then in this case it can fold faster. Okay, now I show this figure. Okay, x axis the contact order and y axis the logarithm of the folding rate. You can see the inverse relationship between the contact order and the folding rate. So, that means what we assumption, okay, what we made, that is correct. If you have more contact order, right, there is less folding rate. If less contact order, there is folding rate is high, it folds fast, right, fine. So, the correlation is also about 0.7 to 0.8, that can see inverse relationship between the contact order and the folding rate. Now, the second parameter, what is the next parameter we discussed? Long range order, right. So, how the long range order is derived? What is the concept used for, right. So, that means the contacts which are close in space, but they are far in the sequence, right. So, in this case we see all the contacts which are close in space, right, and far in sequence, because in this case Nij equal to 1 if the separation is 12 residues. So, here you can see there are how many adjustable parameters in long range order?
Distance cutoff.
Right, one is the distance cutoff,
Sequence.
this one and then this one. You can say distance is 8 angstrom and the sequence separation is, right, that is 12 residues. So, you have two adjustable parameters. We can adjust these parameters to get the long range order, and how far this will relate the folding rates. This is how we get the numbers 12. Right, earlier we used number 12, right. Now, I will explain how we get this number 12. So, we take a set of proteins with known folding rates, experimental folding rates, right. So, you can see the set of two set proteins with known folding rates. So, in this case now we can change the distance of separation, we can change from 1 to 25 residues. So, how many residues which are within this limit? 1 residue or 2 residues, 3 residues and 4 residues, so on. Then from that you can calculate the LRO, because Nij you can put I minus j, that we change, right, 1 to 25, right. For each case we will get the value. So, then we relate that LRO with the folding rate and we can calculate the correlation. For each distance separation we calculate the LRO and we have to get this correlation, that is LRO versus ln kf. For example, if it is 5, right, we get the correlation of minus 0.689 approximately, then you go with the 10. So, you can see the correlation is negative correlation, but it increases with respect to the distance separation up to here 12 or 13, almost the same. After that, if you increase more, then again it is decreasing. So, finally it go up to 25, then again it goes from the minus 0.58. This is similar to what we get from the residues separation of 2 or 3 residues. That means there are some distance contacts which are influential for the folding rates, right. The contact order considers both short, medium and long range contacts, but in the case of long range order we consider only the long range contacts, because that is the one which shows the highest performance. If you add the short range contact, then the correlation is less. That means there is something to do with only for the long range contacts, right. So, from here we get this value 12, this is how we get the 12 residue limit, and with this 12 residue limit we can see the correlation of minus 0.78 with the experimental folding rate. How to calculate the long range order? So, here we give the sphere of 8 angstrom, look at the residues which are occurring within the limit and see the residues, right, which are having the distance of at least 12 residues. So, if you take the 352, I discussed last class, right, how many long range contacts this can make, right, 1, 2, 3, right, in this case 3 by 164, okay, you can get the number, right, for this long range order, right. So, do it for all the residues and finally you divide with the n, right. This is how you will get this. First you do the summation of nij for each of these residues, sum up together and then we get this value, right. So, normalized with the n you will get the LRO. So, now I show the long range contacts, right, at the different structure classes, right, as I discussed earlier, 4 different structure classes, all alpha, all beta, alpha plus beta and alpha beta. So, here in the case of the all alpha, this is mainly here in the 4 to 10 range, and the all beta, so it is mainly in the 11 to 20 range, and the alpha plus beta, right, you can see here, right, in this between the all alpha and all beta, and the alpha beta, right, this is the range. So, this can be reflected in the long range order, because you can see the contacts are different in different structure classes. So, we classified the proteins in 3 groups, all alpha, all beta and mixed class, and we derived the equations for these 3 classes and see whether there is improvement in the correlation. If you do like this, mainly long range order deals with which type of interactions? Long range interactions. So, in this case which class of proteins perform better with long range order? All beta proteins, right, because influence with long range interactions. So, I show the data. So, here if you see all beta, so you can see the correlation is very high, minus 0.92 compared to the all alpha and the mixed class proteins, right, this makes sense. So, if you see the long range order which involves mainly long range interactions, right. So, you see all beta proteins, right, you can get better correlation than the all alpha and the mixed class proteins. So, we derive the equation. So, this is the equation for getting the ln kf for all alpha proteins, all beta proteins and the mixed class proteins. Here y is the ln k, right, and the x axis we can see only the long range order. Now, we compare with the contact order and long range order, right. Some classes you can get the comparable results, some classes this long range order will perform better than contact order. Which class LRO performs better? All beta, right. So, we see the same all beta, we see this very less, it is because they include the short and medium range contacts. That means, short and medium range contacts include some noise in calculating the folding rate. This is the reason why if including these medium range contacts, the correlation is only 0.46, but if you exclude this we will get up to 0.92. So, in the case of mixed class proteins, you can see similar level 0.82 and 0.86, right, the number is almost similar in both contact order as well as for the long range order. So, now we can predict the folding rate, right. How to predict the folding rate? Yeah, because we have the relationship between LRO and the ln kf. So, we have the equation y equal to mx plus c, straight line equation. So, this equation all alpha, all beta and mixed class proteins, So, we calculate for any all alpha proteins, we calculate LRO and substitute the values in this equation, right, and this you can get the ln kf. So, we did this with the LRO, right. So, you get the deviation of just 1.13 right per second. Then we classify the proteins in 3 different groups. We did 4 equations, one equation by considering all the proteins together, right, only one common equation for all the proteins. In this case the deviation is 1.13. Then what we did, so we classified the proteins in 3 different groups, right, mainly all alpha, all beta and mixed class proteins, and we derive the LRO from these equations and calculate the folding rates. If you do like this, then you can see this is the correlation is 0.92 with the different structure classes. This is the deviation, here I give the correlation, right. So, if you do with the classification, you can see the correlation of 0.92 for the different structure classes alpha and beta and mixture class. So, here show this figure, right, show the comparison between experimental and the predicted ln kf. So, you can see there is a good correlation between the experimental and the predicted folding rates, where most of them are very close to this straight line, right, this slope. So, we discussed about the contact order and the long range order. Contact order considers the distance of 6 angstrom and take all the distances. Long range order we have one more conditions based on the distance separation.